In this video, we will learn how to run co-occurrence analysis in Dimensions LNC, a powerful tool which enables you to generate insights from texts of hundreds of millions of scientific documents within seconds. We will explore co-occurrence analysis general use cases, as well as specific ones, for which there are pre-built tabs in the tool. Finally, we will see how to export results from the platform to share them with colleagues and save an analysis in case you want to rerun it again later. Co-occurrence analysis is a quick way to start generating insights from the full texts of hundreds of millions of documents. You can find it in the menu bar under the Analytics tab. In this tool, there are 4 different tabs available for exploration, for each of which you can hover over the eye icon to read more details. The Correlations tab allows you to correlate concepts of any nature from our ontological tree. The system will search for instances where these two concepts are often mentioned in close textual proximity, which usually means that those concepts are somehow connected to each other. The system will run this terms co-occurrence analysis semantically across hundreds of millions of documents within seconds. In the Correlations tab, you can select any concept on the left from any knowledge domain that we cover. And the same for the right. You can simply type it here in the search box and select a term from suggestions. Or you can use Domain Explorer to identify the concepts that you want to correlate. In this ontological tree, there are 40 million concepts structured into 23 knowledge domains, and any of them can be used for correlation, including cell lines, chemistry, companies, devices, diseases, drugs, processes, genes and proteins, polymers, substances, etc. Alternatively, you might be interested in exploring these 3 pre-built tabs: Correlate Organizations, Correlate Mechanisms of Action, and Correlate Pathologies. The difference between these tabs and the Correlations tab is that they have pre-selected subsets of domains or concepts that can be correlated. Let's look at the Correlate Organizations tab. By default, the term you include on the left will be correlated with the preset All Companies on the right side. You can also choose from the other presets, Top 50 Pharmaceutical Companies, or Top 50 Chemical Companies, or Research Institutions. And on the left, you can select from the domains that are listed here. It could be a drug, a disease, a device, a chemical compound, etc., but not any concept from the ontological tree. Similarly, in the Correlate Mechanisms of Action tab, on the right you have presets, such as pathways or genes and proteins. And on the left, you can select either a disease, a drug, or a secondary metabolite. Finally, in the Correlate Pathologies tab, on the right you have the option to select from pathological processes or toxicities. And the domains that are available for exploration on the left are chemistry, diseases, drugs, natural products, and polymers. But the idea is the same as the Correlations tab, and you simply start typing on the left. For example, a disease of your interest. If you want to explore pathological mechanisms of a specific disease, select it from suggested terms and simply hit Search. When you click Search, the system generates this graph visualization. Here on the left, you can see the selected concept. In my case, bipolar disorder with its subtypes and children. In the middle, you can see specific concepts that were predefined for the analysis. In my case, pathophysiological processes. And on the right, you can see the list of original sources containing the terms in close textual proximity and where potential connections have been identified by the system. Importantly, you always have a link to the original source, which could be a publication, a grant, a patent, or a clinical trial. This allows you to dive into more detail and explore exactly how these concepts are discussed in the text. In the middle, you can explore these concepts, which in my case are pathophysiological processes, by clicking on any of them and asking the system to perform several actions. You can load specific sentences. You can refine the search. You can show concept details. You can temporarily block it or add it to a block list for future searches. If you click Load Sentences, you will see all original sources where the potential connection was identified on the right, same as before. and the snippets showing you exactly how the two concepts are mentioned in the text together, so you can start generating hypotheses immediately. Alternatively, you can load details of the concept if you want to understand the parents or the synonyms. And as mentioned before, you can further refine the search to your needs. For example, you can select to focus just on bipolar I disease, and then the visualization will change according to your specification. You can block any terms that are not of interest to you. For example, general terms or those terms that you already know, using a temporary block. Select Block, and the system notifies you that there is a blocked concept, which you can always unblock if needed. You can also create a block list to use for other searches and analytics. Create it from here by clicking on a concept and adding it to the block list. On the left side, you can explore different visualizations. One alternative visualization is to see the top pairs table. In my case, everything on the left will be bipolar I disease because I specified it. You can ask the system to show top 50 concepts on the left, which in my case does not make a lot of sense because I have narrowed it down to one disease. But top 50 concepts on the right makes a lot of sense in my case, as I can see the list of all these pathological processes connected to my disease with a frequency of co-occurrence in texts, with the most popular or the most frequent terms on the top by default. You can use this box to filter down to specific processes, like, for example, inflammation. You can jump between pages here. If needed, you can further refine your search if you want to focus just on specific repositories and include some of them. Or if you only want to review recent concepts, for example, those concepts that were recently detected for the first time and stored in our system. And from here, again, you can manage or create a block list when needed. To apply these changes to the settings, I would need to rerun the search. Finally, if you want to export this graph into an external slide deck or share with your colleagues, you can download it as an image. You can also save a search if you want to repeat it later by simply using this Save Search feature on the top. In this video, we have learned how to generate insights from texts of scientific documents quicker using co-occurrence analysis. The pre-built tabs correlate organizations, correlate mechanisms of action, and correlate pathologies can be used for specific use cases to identify chemical suppliers, understand disease or drug mechanisms, and identify potential drug targets, among others. To get the most out of Dimensions LNC, please watch additional videos dedicated to specific features, analytics, and use cases.